to Andra and Lynn for doing a lot of
the. Are you recording? Sorry, she didn't record yet.
Yes, I hit record.
Sorry,
the moment you said hit record, I hit record.
Okay, go ahead.
Okay, great. Yeah, and thanks for everyone being here, Andra and Lynn for doing a lot of the organizing with this, and Sue and Nicole for also helping on the Biocuration side of this. And so today we're going to introduce to you Gene Wiki, and hopefully you will see, if you haven't already tried it out, some of the utilities from Gene Wiki, you'll see that there's a lot of useful applications, and we'll go through that today. If you want to follow along with us on Twitter, we each have Twitter accounts. I think Andra linked his in the main slide, but you can follow the hashtag here. For the workshop, let me reshare my screen here to desktop 2. For the workshop, here's the link, and I'll put this is all in the chat. So if you're not already on the Slack channel, I think there are some people that already joined just now. If you're not already in the Slack channel, definitely be sure to follow that. And then here are some additional links. So one link you'll want to know is the program. So that's what I've clicked on here. And then through the program, you can see we have these 2 breakout rooms. We do want to know what people are signed up for, because in order for us to assign your breakout room, we need to be able to see what you've requested. Otherwise, you won't have a breakout room. So that you can do right here. That will take you a couple of seconds. And then if you want direct access to the slides, you also have access to that. Again, these are all in the Slack channel, but just to put them here for you. And if there are any additional questions, feel free to ask them in the Slack or the Zoom. I'll be kind of moderating as Andra and Lynn do the presentation. And with that, I'll turn it over to Andra.
So,
and then if people wouldn't mind, if you're not talking, unless you have a question about something, maybe no video unless you might be the speaker.
Yeah.
And please mute if you're not speaking, just so for background noise.
So welcome today. I will start with a brief introduction of the Gene Wiki project, where I'm collaborating with Lynn and Saba and others. And in the Gene Wiki project, we are leveraging Wikidata as a central infrastructure for life science data, and it's as a hub for linked data to other linked data databases. So what is Gene Wiki? The Gene Wiki project started in 2008 with the initial aim to create a Wikipedia article in the English Wikipedia for every human gene. This was done by creating the Wikipedia pages by adding 2 parts. First of all, the info boxes. The info box is a part of a Wikipedia article that presents and renders structured data that is imported from various databases. So in this example, you'll see different identifiers, expression patterns, and that's nicely rendered in the info boxes. And at the same time, add summarized knowledge from the references, etc. So the idea was and is to, by creating stubs, the Wikipedia community starts enriching Wikipedia for those specific Wikipedia articles. So this is an example of such a Wikipedia article of a readlin, and which is a rich source for additional knowledge. However, as trying to extract this, the expression of the protein has been found to be significantly lower in schizophrenia and psychotic bipolar disorder. It's very straightforward and easy for a human reader. However, to have computer analysis or to ask more complex queries, extract more complex information, which is in Wikipedia, such as other proteins that are involved in the same disease, it is really, really hard to get that information out. Another thing about Wikipedia is it's a large, interconnected, but independently maintained network of 300 language communities. And the information is not evenly distributed over the different language communities, versions. So here I have an example of the ICD-10 code for malaria. And depending on the language that you're assessing, whether it's Dutch, Greek, English, Japanese, or sorry, that's an error, it should be Hindi, you'll get different numbers. So the Dutch Wikipedia only mentions 50, B50, whereas the English Wikipedia and the Japanese Wikipedia mentions B50 to B54. And trying to synchronize this across the different 300 language informations takes time and effort, and needs to be done by the independent communities that drive the different language versions. So in 2012, the Wikidata emerged, which actually today, 8 years ago, because today is Wikidata's birthday, which is like it's from the same organization, the Wikimedia Foundation, that hosts Wikipedia. And it's similar to Wikipedia in the sense that what Wikipedia is to text, Wikidata is to data. It's completely free, even for commercial usage. Anybody can contribute. It covers all domains of knowledge, so not only the life science, but also history, cultural, GLAMs, libraries, museums, you name it. It has an extensive item history. It integrates well with the semantic web. It's stable, where research projects have end times, which also sometimes dictate the availability of the data. The data can live on Wikidata, and it's actively developed, and there is a large number of editors. So now I'm going to give a quick demo of the infrastructure of Wikidata, starting with a Wikipedia article. So this is a Wikipedia article of a mutation of the brand gene. Now, if we want to go to the Wikidata item, there is a link here in the left. By going there, we're entering the Wikidata item. The Wikidata item then again can be structured in three sections. There is the header section, which contains multilingual language descriptions and synonyms. Then there are the statements, where the statements are derived from various primary resources. And there are the links. So for this mutation, there are 3 Wikipedia articles in English, Farsi, and Norwegian. If we then go to, so now we're entering the links to, no, I'm going too fast. So if we want to now browse to other missense variants, we can, by clicking, can explore different other missense variants and see that the Wikipedia article is missing here, but there are links and links to other databases that can be explored that has information on these gene variants. And this information is then also available in Sparkle queries. So we can, for example, start with a cat. We have a quick query here, and you can make it as complex as we want, and we'll demonstrate some more complex queries later in the program. So what Wikidata offers, it started as a knowledge graph for the Wikipedia articles, but now has a merit on its own as a hub to not only Wikipedia but also other databases, and that data and that knowledge is available in a unified way. So now moving forward. So that's the end of my quick introduction. This is our latest paper, which is available online, and we have to thank everyone involved here, as well as the thousands of Wikidatians. And we have some links here that I suggest to discuss. And now I want to hand this over to Lynn.
Okay. Sorry, did you hear any of that?
No.
It made me muted and unmuted. It kept flashing. Okay, I'm sorry. Okay, here we go. I was saying I'm really happy that the Biocuration community can come together for this meeting, that we found new inventive ways to reach out to each other and to share information and to continue working together, you know, despite what's going on in the world. Today I'm going to talk about briefly how the disease ontology, as one of the many biomedical ontologies that are out there and that we're familiar with, teamed up with the Wikidata team. We're on our second R1, so I've been doing this for a bit, to really integrate additional curated knowledge into a Wikidata to enhance what the Gene Wiki project had already been doing. And so I'll introduce you to that, what we've been doing with that, and then just really briefly the symptom ontology, which is one of the examples we're using today. And the reason we chose that is it's a part of the umbrella project of the Human Disease Ontology project. It's also CC0, and it's a way for us to augment the information we've already incorporated into Wikidata through the disease ontology. All right, let's see if this will advance. All right. All right, sorry, I'm trying to get to advance and it's not. Oh, okay, it did. All right, good. All right, maybe just a little delay. Oh, on, okay. All right, cool. All right, here we go. So for the disease ontology, if you're not familiar with it, it is an open biomedical ontology for the domain of human disease. We're a genomic resource for NHGRI, NIH. We've been working on this project for about 15 years. The reason we teamed up with Wikidata in this case is that it was a domain ontology. It was a concise project, and genes and disease was the type of data we wanted to connect. One thing to mention is that we utilize both oboe and owl trees in the ontology world, which means we're able to have a description of just diseases, but also from this little pop-up window you can see, use other biomedical ontologies, importing them so that we can see, add additional information about diseases based on this other information. Say in particular what phenotypes are associated with what diseases, for an example. Okay, and then I'll go on. Great. Okay, so for the Wikidata project, what we want to do is have highly curated data that would also that was documented well, so that we would know the provenance of the data. So we did it for both drugs on the right-hand side from PubChem and Drug Bank, and the disease ontology. We wanted to be able to set up systems so we could automate the QC, automate the loading, and bring all of that together with genes into Wikidata. And the reason community is on this slide is that community played a pivotal role. If you haven't worked in the Wikidata, Wikipedia community before, you may be surprised to learn that the community is a big aspect of it. So it's not just the people that are on the particular grant doing the work. The community has a lot of input. There's a whole proposal stages where they give you lots of great feedback, but also the community has a lot to do with the data that's there. So I think it's an integral part of that, this aspect that's important to note. Okay. Great. So data modeling came first. So we knew we wanted to connect these types of data. How do we do that? We looked at different relationships between these concepts and then modeled all of that out. We tested it with some initial data. How would it work? How would it come together? And this is the same sort of format we've done for every type of new data we've brought in subsequently. We really want to model it out, figure out, does it biologically make sense? Then how do we bring that into the Wikipedia space? In the Wikidata space, rather, so it makes sense in that system as well. So it's just kind of one of the initial steps you do. Okay, great. And I'm showing you this slide again, just to really show you the diversity of information of what we wanted to bring together. So as Andra was saying, and we'll talk about even further, we have genes, diseases, and drugs, but how do you link them up? Right, so we had to bring that sort of information together, but then we had to automate that information by what we call bots, and they'll be talked about later by Saba. Really, it's an automated system that queries the data, does the QC, brings the data in, and identifies curation sets that we have to do. So in this case, it's one nice thing is you can have a very enriched data set. As you can see for COVID-19, I only give you a bit of the page, but the number of articles in different languages is quite, over 140? My glasses aren't wrong. Yeah, over 140. And then you can have all sorts of different ID space you can link out to it as well. So it's a really growing, energetic kind of system. Okay, and one other point I wanted to make about the languages is that it's really provides access to many different communities. What we found over time, and this is a figure that Andra had put together this last year, is really giving you a sense of the number of terms in each language. And this is a little bit out of date, so it has grown, but different communities can have interest in an area of Wikidata and then add those translations. It really does add to the larger community's information. And I think it's as an ontologist, I didn't have to translate my terms. They were translated by the community. So it's really an enriching activity. Okay, so a little bit about the crowdsourcing aspect. So beyond getting the initial bot in place and getting the data integrated, going through those steps, a little further along in the project, we sought out what data was coming in through Wikidata from the community itself. Because people, you know, as we'll show in one of the breakouts, you can add data, well, anyone can add data to Wikidata. You create a login and there you go. But there's quite a bit of information coming in this way. So on the Wikidata side, there was Sparkle queries written to capture this information, do some QC. We built Jenkins instance so that we can have reports every month of what the new data is. And that allows the curators on the disease ontology project to then assess this information, but it's a very targeted set of curation. I found it very useful. We've added thousands of cross-references in this way, and it's an ongoing process. So it's very beneficial to the ontology and then also really capturing the information from the Wikidata community. And it's fed back through the system as well. Lots of QC, which is good. Okay. And next couple slides, and we're almost there. So another thing I wanted to show you about exploring disease, Andra had a slide showing you about the Wikidata query service. Now this is a fantastic service that allows you really to mine and play with the data that's in Wikidata. And there are a number of those pre-set up queries, like Andra did cats. This is one of the disease ones, looking at infectious diseases, and then their various drug disease interaction information. But the reason I'm also pointing this out is I want to expose you a little bit to Sparkle. You don't have to become a Sparkle aficionado, but just say if you wanted to change the language in here, you can see in the middle of this code, there's a little EN in quotes. You could change it for the code. Maybe you want to do French, put a little FR in there instead. It allows you to play with the data, I think in a very safe way, and then explore the information. And then one thing I'll show you next is just a little bit of the little blue button to execute, is just a little bit of the output. It produced a network graph. This is the result. I just pulled out a piece of it in a screenshot. But this is showing you the diversity of data that's associated with COVID-19. If you just take a look around and kind of go a circle with your eyes, there's a lot of different information that's associated. And I think you get more of a nice visual this way, more than just the lists we show you. But it's a very rich data set, and it's continually growing, and many sources of input. Okay, and just last two slides to introduce you to the symptom ontology. This is an ontology we started 15 years ago along with the disease ontology project because we wanted to have a rigorous way to collect symptoms. So over time, it has it's been pretty steady. We've been integrating more information. But one thing I want to point out on the ontology side, the screenshot I'm showing you on the right in the blue box, this is from the disease ontology website. What these imports allow us to do is to have another view of disease. So this is the view through symptoms. So this screenshot is showing you at least a partial list of all of the diseases that have the symptom pain annotated to them. And so this allows another way to look at information from a different perspective, but all within the ontology itself. And there's links there, and then you'll have the slides if you want to explore. And then just one last slide. Wanted to explain a little bit of what it took to bring in the symptom ontology for this workshop. So this was new. Andra, I think, proposed it in the spring, this would be a good example for a number of reasons. It's CC0, which is what we want in Wikidata. It had the structured information that's associated with diseases. But one part of the process I'm pointing out here is this human community aspect I pointed out earlier. You have to make proposals. It's pretty well outlined and straightforward in the Wikidata system how to do it. But this is where the community comes in again, to help you model it, to give you feedback. And then anyone who has an ontology who's interested in doing this, we're happy to give you feedback on how to do it and pointers. But there are a lot of groups requesting ontology space. ontology ID space in Wikidata. So I think it's really grown over the years. So that's it for me. If you have questions, please let us know. Do I have to turn it off sharing?
Yeah, you could, yeah, stop sharing.
Yep, got it. It's all yours.
And then, yeah, so thanks, but we're really good on time. That's great. Yeah, so hopefully these were helpful for both of you, just as for all of you as an overview. And thanks again to Andra and Lynn for doing that. So here's where, oops, this is the wrong one that I want to share. Here's where we are in the workshop now. So we just went over the 4 aims and then the introduction. The first one was by Andra. The second one part was by Lynn. And now we're going to do breakout. So Lynn is leading workshop 1 and Andra is leading workshop 2. These are already set up for all of you. So this will be over the next 60 minutes and these will not be recorded. So that gives opportunity for people to have more open discussions. I'll be here in the main room. So if you ever have a question about something, feel free to just leave. You can leave your breakout room and come back to the main room, and then you can join your breakout room again, and that will happen over the next 60 minutes. So I'm about to open all breakout rooms, and then we'll come back, and then after the breakout room, we'll have a 10-minute break, and people may be ready.
We are recording.
Okay, great. Thanks, Sue, and it's great. Seems like people had a good set of breakout sessions, and this is a time where people can have their video on if they'd like, and their mics off, so we can just have, we have a little bit of extra time here, so this can be kind of an open Q&A and discussions of things that people gained from the breakouts that they thought were good, or things that they'd like to see more of going forward as we go on in the discussion forums. What did people think of the breakouts?
I just wanted to say something. Is it okay for Lynn to give us an overview of what was discussed in breakout room 1? Because I was in breakout room 2.
Sure. Yeah, I could do that really quick in a few minutes if you like. So we went through the steps to how you would add an item to Wikidata. In the chat window, I'm going to give you the Google Doc that I used. Oh no, sorry, that's the agenda. I got to go one more over. Apology, I changed screens. But I'll give you the link for adding, we just add symptom ontology to Wikidata. And these steps in the document I'm posting now to walk you through it. So where you check it, does the data exist? What properties you use to add it? How do you query Wikipedia and Wikidata? Andra put together a really nice notebook to, you just click on the steps and you walk through it. And then it just shows you where things are pre-populated and then what information you need to document and put things in.
Just Lynn, one note, the symptom list, I had to send you an email to request access.
I changed that.
Okay.
Yes, sorry about that. It is now open to everyone. Yeah, I didn't catch that till after we were done. I saw my phone then. Thanks, Sue. You don't really need the symptom list. It was an extra backup, but yeah. If you want a list that has those, some of them defined. So.
Does that answer your question a little bit, Arwa, or are there additional questions you'd like to know about?
No, that's it. Thank you.
What else? And I just linked what Andra put in the Slack channel too on slides here. What do people think of the breakouts? Are there things that people really liked? Are there things that people would like to see more of going forward?
Would you want to be suggesting types of data for us to add to show you how to add it other than what we did? Other information more pertinent maybe to your work?
So hi, this is Sabrina from ZFIN and the Alliance. So for me, there was the very first time I saw anything about Wikidata. So it was really, very instructional. I think one point that we have discussed is how to utilize all the data that exists at the level of the animal models, and how all that data really, for the best of the community, really should be included in this. And as we discussed, this also requires funding, which is very hard to come by nowadays. But I think this is something that that it would be good to keep in mind, because I think there's a lot of very good information and very good information and a lot of data out there that could be regrouped in one single place.
Are there, that's a great comment. Are there certain examples, like when you're saying that they're data from animal models, like what would be one model where you think certain data sets would be really good to integrate as part of Wikidata?
Well, I think we sort of mentioned animal models for diseases, and I think that these exist in pretty much any animal that is used in research. So thinking at the Alliance, we have mouse and rat and zebrafish and C. elegans. So this is the first example that comes in mind, but I'm sure that there is much more data that could be added.
But
Yeah, and I put it's disease models I think you'd want, not diseases.
Yeah,
not diseases of those organisms or models of human disease. So.
Yeah, it's not a mouse disease. It's a mouse that models the human
Yes,
disease.
yes.
At least for us. I mean, there are other groups that do handle human disease, or do handle animal disease, but when Sabrina's talking about animal models, it's really the model organism database data that it would be helpful. And I know Cindy mentioned that the MGI is talking with Andrew about trying to figure out how we create those links, because it is something that we don't necessarily want to have to hand enter everything that's in MGI, but also looking for better ways for community users to enter data into Wikidata that then can get picked up and pulled into the model organism databases.
Great. Additional thoughts?
One point the camp in our session was talking about how users from other language Wikidata communities, how are they contributing when their data is entered? Has it contributing back to the English version of Wikidata? Like, what's the role back and forth of sharing information? Because we know, so one of the people, Sabrina, noticed that the OMIM we're in one maybe language, but not in another's records. So how's that shared, and is there a feedback loop? And I think interest in how Wikidata may be helpful to groups to do translations or to facilitate that information. The sickle cell ontology came up as an example.
Isn't there already something in place? I remember from the COVID hackathon, Andra, was it you or is it Egon who worked in the translation bit a bit as well? And there's like well, maybe not a bot, but there's another tool that translates things.
Yes, there was indeed in the biohackathon. There was a topic, and I think that was led by the Japanese team, who are depositing their data, and they have I think that was on imaging. So they are using they have images with indexes in Japanese, and by depositing it into Wikidata, aligning it with Wikidata and the different models, they were able to. You can now actually browse their. You can now browse their images, and which is a Japanese data. I think it's Togo. I think it's togo.tv. I need to look that up. It's from the Tokyo University. So they were depositing that into Japanese, and then it's now available to English. But we didn't get to ontological mapping between English and non-English ontologies there, but it should be straightforward.
Yeah, but someone had some sort of translator tool. I think Egon was involved in that. I think he was dipping into that channel at times and helped them.
Could be.
So there is, I'm no expert as you can hear, but there is something already going on where people are trying to translate things and map things up in different languages. Yeah.
There is one caveat there, and that is Wikidata only covers 300 languages. So that's still a bit limited to, I think there are 7,000 that are still yet to be covered. Yeah, it's a very valuable tool for translation into multiple languages.
Great. Other comments and thoughts?
Yeah, I've got a more generic question. Something that we haven't covered is how widely is Wikidata used within, like, I think we're all coming from a biology background here. What sort of projects are actually utilizing the Wikidata at the moment? Because I'm involved in getting the complex portal data in, but we're not yet sure who will use it except for, well, Wiki Pathways will use it. So we've got one use case. But where do we have successes already?
That's something
It'd be
I
nice
will talk a little bit about in the next section. But I don't know if Andra and Lynn, you want to add besides the ones that the examples I put in there. I mainly have those 3 main examples later.
So there is we did I think 2 years back we tried to assess the usage of Wikidata, and but we looked at that point at the queries that were asked, and I need to pick that up. There is one weak spot of Wikidata, and that is being which is a great point and a weak spot at the same time, which is the license. So the CC0 license is really great because it prevents license stacking, which is problematic if you want to integrate various data sets, and if you have to compile different licenses, it will become really complex to do. But the CC0 it doesn't require to attribute. So we are attributing with references, but then we have no means of accessing the queries. But we did some initial studies where we looked at the combinations in the Sparkle query. So let's say there is a disease ontology ID and a symptom ontology ID and a gene ID. We can say that the data is being reused in those contexts. But then the European privacy laws kicked in. So we had to stop it because we are not allowed to use that data because Wikidata, those log files are in Germany. So we can't assess that. So it's the only way to get access to where is Wikidata uses, like you just said, Wiki Pathways is using it, you from the complex portal we're doing, I know BridgeDB is using it, but I'm not aware yet of a systemic search, so I'm open for suggestions there. Maybe also, I don't know if Thiago is still on the line. I know he was running the COVID project on Wikidata. If you could elaborate a bit on the reuse, use, and multilinguality of the COVID project there.
Sure, I was half on the line. I was solving another issue. Sorry. So in the COVID-19 project, I don't think we did any kind of like concerted effort to get it multilingual. It was mostly that many of the editors are not originally English speakers, so there have been edits, but not really. So I don't have much to add, though. Sorry. Yes.
Maybe this can be a continued discussion point. We can have like a thread or something going in the Slack channel. That could be a good conversation. Yeah. Great. Well, it seems like it's just having some good discussions and good conversations, and that people are getting useful things out of the breakouts, and we can continue on the Slack. And with that, I will go over the methodology, and so kind of applying the things that you're learning as you go forward with, as you get familiar with the steps of creating a Wikidata account and then starting to do entries, manual entries, and the process of wanting to automate that as you work with larger and larger data sets. And maybe I'm a really good person for this because I've only been doing this myself for about a year. And so I feel like I've really learned a lot from Andra and Lynn and the whole group, GeneWiki group. So maybe I'm a good person for people to ask. I think there was someone that asked about programming experience. And so yeah, myself coming from no background basically of having written a bot before joining this group, I can say that it can be done. And I initially learned how to do it in, I think, my first month of working in the group. And now there are a lot of things that I have learned in that process. So hopefully these will be helpful discussion points for all of you. And of course, if there's anything else that people want to add from their expertise, feel free. So why and when for creating a bot? I know that Andra walked through a little bit of this already in the breakout, so maybe some of this is repetitive, but it's hopefully good to reinforce these things. So I'm going to give the example that I have mainly worked on, which is the ClinGen bot example. So here is my history, and you can see my name here with the account where I switched from basically my name and manually entering, when is this, 2019 of October, and then when I got it scheduled on the bot. So I think I formally got it automated into a bot form in November of 2019. And then you can see these updates with the protein box bot since. And here's the link to, again, the slides are in the Slack channel if people want to see this directly. But here's the link to where you can see all of our scheduled bots, specifically for the Sue Lab and GeneWiki project. So here's a bunch of them. Some of them are working, some of them are not. But here they are, and here's the ClinGen one here. And so going forward, why would you want to do this? Well, hopefully this is pretty clear, I think, by this point, but you know, as opposed to, as you can see, and we'll go a little bit over this history, as you can see, as opposed to doing, you know, maybe you just have quick things you want to edit here and there, and it's a quick fix that will take a couple of minutes, but then over time, as you get more and more data, then you're going to want to have that automated. So maybe if you have just a few entries that you're doing by hand and with your Wikidata account, you know, maybe that will take you 10, 15 minutes to do those by hand. But then in this case, so you can see the most recent successful run, and you can click on this here and it'll take you. So now you can see that it's not running very well, but there's another one running, and we'll go over that in a little bit. But this took a total of 24 minutes to run, and it's over thousands of data entries from the ClinGen database. And so what's happening actually is it's taking data from, well, we'll go over, from the ClinGen database, and then it's automatically updating that into GeneWiki, or sorry, Wikidata. And here's another example with Celosaurus. So Andra is involved in this too, and so this is a tweet that includes Andra. But here you can see, you know, it's over 100,000 entries that are now part of Wikidata. And, you know, Lilia and wrote the bot, Andra did a lot of help. So basically, you know, being able to write a bot, the first run of it takes a bit of time, but then it's much, much faster to do this and integrate whatever data sets you're interested in having be part of Wikidata than doing this manually, even maybe in your own research. And so with this example here, basically I wanted to show that this bot I originally started running, like I said, in November 2019 of last year. And I'd say there's been two main points where there's been trouble, and here's one of the other major points. And actually, so if I go back to this and we look and see, so anyone can click on this and see what happened. So this is, I clearly have to fix this, but if we look and we want to see what happened here, so this was the last, says last successful build versus the last build, and then I made a little output data file set, so I can share that with you too. If I can open this here, my downloads. So open this. Here we go. Let me share that with all of you. So I made this output file, which is, you can think, analogous to a log file. That was just when I was starting to get familiar with the data. And you can see here, so this is the file that it makes every time there's a run. And of course, I'm going to be making some edits to this now since I've done it. But basically, if I'm going to zoom in here so people can see this a little bit better. Okay, so this file, basically I get my input file from the database looks a little different from this, and everything from here onwards is what I've added. So in my script, I set it up to walk through each of these lines. So here I have my gene, and then it kind of goes through, you know, which number it is, and I guess it's starting at 0 here, which is fine. but I give a note here if it worked or if it gave some error. So it's just binary as error or complete, and you can be more detailed with this. I've definitely done that. I think there's also some where I put updated. If I search here, wait, maybe it's not in here, or new right or something. Anyway, so I have my gene. Here's my GNC gene ID for that gene. Here's the disease name. Here's the Mondo ID here, Mondo ID. And then here's the standard operating protocol. And here's how it's inherited. This was the first time, I think in April or so, March or April, this was the first time I had a problem come up. And that was just because ClinGen added this column in their data set where there was only, I think, 9 before, and then there were 10, and now they've actually reduced it again. So that's that's why I'm having it not run anymore, or why it's not running anymore. But anyway, so going to this section, this is my output file to kind of help me debug what happened if there's an error. So it will say, so I have some criteria that I've put in here, and then I have this kind of confirmation of what the end QID is for either the gene or disease, if it worked. And so you can see here, if it's an error because one of the criteria is it has to be a definitive gene, and then also it has to be definitive classification, and then also there's no QID for that disease. And so there's also things I put in there to write a disease. So it will create a page, an item page for that disease or for that gene if it's absent. But if it's a definitive classification, it won't write the rest of it. So here in this case, you can see it worked, and that's because there is a page for both of these, and also it's a definitive classification. So going back to this example, if I go back to the slides here, yes. So going back to this here, so you can see what happened. So the last time it was working well was early September, and I've been doing a lot of other stuff in that time, so that's why I haven't gotten around to it yet, but I will be soon. But if we want to see how this works, so it's like last unsuccessful build, last successful build. So if I want to see what happened in this last run here where it didn't work well, I'm going to go to council output, and probably Andra already went a little bit through this. So you can see that things are happening here. I'm walking through, the timer is started. I put a timer in my script, and it's connecting with a protein box spot, which is what we have to run the job for this bot. And then it's going through and it's collecting different things here. It's going through pip, cloning from the script from the GitHub, so that's over here. And then as it's going through and writing this, copying, etc. So okay, let's find the error that happened. Okay, so I'll always give you this output, and this is where you're going to find your error. So let's see where the error comes up. Here we go. Trace back most recent call. And so it's basically saying that here there's an issue with the number of columns and that's not matching up. So value error, length mismatch. So basically the new values have 9. So what this means is that ClinGen updated their input data set to be 9, and before it was 10. And like I said, the last time I had trouble with this, they had made it larger. So it went from 9 to 10, and now they've gone back and made it from 10 to 9. And so this will always happen, and it's expected. Of course, you want to go and make your script as sustainable as possible, but these are things that are kind of outside of your control. And so you have to go back and adjust for these, right? And easy ways to figure out, so what does this mean for those who are new to programming, like myself? I did a lot of R before, and now this is all Python. So things you can do is you can just type in value error, and you can start to look in, okay, what does this mean for a value error? So maybe this is repetitive for most people, but there's a lot of stuff that, and then it sends me an email too. So now it's saying that, okay, we emailed you to let you know. So I've basically been getting notifications saying, okay, I need to work on this. But I thought it would be a good thing to walk through as an example that, something that can go good or badly over time, and kind of how to continuously work on that. And going back to this council here, so if we click back to this, so you can see these are the main bots we have going, and probably Andra already walked through a little bit of this in the breakout, but this one is running right now, and then they'll be in queue, so then it will try again. This is on a weekly schedule, and then it says here how much time it runs. It usually takes about 25 minutes for this one because it's not a huge data set. Okay, so going back here, so there's always going to be issues that you'll experience. So this is already a year out. I created this script for the spot and got everything together. I think it took me a total of 2.5 months, 2 months, having never run before. But once you get better practice, it probably will take you about a week or two, and then you're kind of getting feedback and going through the process. So what does that process actually look like? And also, as I'm going through this, please, feel free to use the chat to just ask questions freely, and I'll see those pop up, or if you want me to slow down, and I can go and walk through those as we're going forward. Okay. Any questions so far before going through this process of how I started to put this together and kind of the things that have happened since? Okay, so let me just do this. Get rid of these things here. Okay, so where and how to begin? So again, any questions up to this point? All right, so where I started, and I think again, Andra and Lynn have probably already commented on these things in the breakouts, but the main tools I use are a Jupyter Notebook for just drafting my scripts, GitHub, because that's what our group uses is GitHub, and then community feedback, right? So a big thing that's been emphasized a lot throughout is community feedback, and and you'll kind of continuously go through this cycle. So, I mean, me being the person that I am, I was like, okay, I'm going to get this done. I'm going to make a good impression in the group. And as I think Andrew has said, it's really that last 10% that ends up taking the most time. And it's totally true. The year after, there's still things that pop up, and that's fine. That's the nature of doing these automated bots. And really the community aspect helps a lot. So anytime we have meetings about once a month in the Gene Wiki group, and it's a really good space, really welcoming space. Both Lynn and Andra and others in the group are very welcoming, and it's a really comfortable environment to talk openly about things and get feedback, which I appreciate, especially during these times. Okay, so where did I start? So me being a total novice, I had not made a Wikidata account at all, so I might be in a similar place as where some of you may have started a year, where I was a year ago is maybe where some of you are starting now. So if I go to this history, I'll click here. So this is one example. And if you notice from the Excel, that was the very first one that popped up. So I'm already logged in here as Saba. So that's my account here. And you can see in this view history, so if we go again to the item, original item, you can go to view history, which I know might be a little repetitive here, but just to reinforce. So you can see where I first started doing things. So, okay, Andrew did something here. We had a bot over here. Now I've started to work in the Gene Wiki group. Andra probably helped with something here. And we can see exactly what those are if we want to. We can compare this and this, and we can just say, okay, let's compare these versions. And then we can see what exactly those were. Oh, I need to do a different one actually. We want to do, I don't want to do that, but that's okay. Yes, so you can see where I am manually doing things, Andra is manually doing things, Andrew's doing things, our bot is doing things. And then there's a switch, right? So then you can see here that I was doing a lot of stuff in October and September, it's very productive. And then I switched it over to it being automated. And so that's where the protein box bot takes off, and that's kind of where it's been happening now. So what's been going on? So anytime, this is basically doing updates to this specific gene. So if everything is fine in the script and this doesn't need any updates either on, this is the gene side or on the disease side, then everything is fine. So actually let's go to the disease side. So let's go here back to the item. So what is the actual thing happening? So I mainly worked on this section here. So that's what this bot does. The ClinGen bot that I have is just doing, looking at, okay, what's the disease association between the gene and the disease? And so in this case, one of them is Noonan syndrome, and you can see that that was put in here in 2019. I probably did that manually as a test, another one in 2019. Another one, October 2019, and then where are the updates instead? And then here's the references. So I can then click to Noonan syndrome, and I can look at the history here. That's the association. So making sure that there's symmetry on both sides. So again, you can see the edits that I had, and then Andra did something, and then protein box bot did some update here probably. Okay, so this is, so first just walking through, and when I was first doing this, again, going back to that A2ML1 example, when I was first doing this, as you can see, I was doing it manually. So I would just edit something and I would just say, okay, state it in. I could make another reference here if I wanted, and I could say, you know, state it in my property. I'm going to do clin, and then it will autofill the rest. So maybe I want to do clinical try, whatever. I want to remove this because I don't want to mess this up. So I'm just going to do cancel. But that's an example of how you would write something in manually, which I think has already been, you've already walked through a little bit of that. So I tried that out manually, and I had this whole data set, I think at the time was around 600 rows or 600 entries. So 600 entries, meaning that there's a gene and there's an association. So about 1,200 max, if you think of symmetry, but probably less than that because there's repetitive scenarios between genes and diseases they're associated with. So I first just started to make sure that I understood the problem and I understood the data of what's going on. So I didn't really, my background is actually microbial ecology for my PhD. I did do biomedical research during my undergrad and a little bit in my master's, but it had been a little bit since I had familiarized myself with biomedical data. So first I needed to kind of look and see, okay, do I understand what these genes are? Do I understand what these diseases are and how they're being written? And you know what sources they're coming from and why they're coming from those sources, and the codes associated like HGNC and Mondo and things like that. Omen was one that came up in our discussion. So just understanding what's going on and just exploring that a little bit by doing things manually. And there is a sandbox option where you can test things out in the sandbox, and that's also really nice. And that's the advantage of using the sandbox option, is nicer because then if you write something and you write it wrong by accident, then there's no consequence. In this case, you know, nobody else is really doing any of this except for me at the time, so it's okay. But realistically, you want to try stuff out in the sandbox, and then you can move it over, because once it's published in Wikidata, you know, it's published and you have to go back and change that later. And maybe, you know, in the time that you're learning about stuff, you know, somebody uses that entry for whatever reason, but usually not, but it's a good practice to use a sandbox instead. And so because I was coming from a background of using R, so I was manually kind of playing with what these associations were and editing them and adding new ones just to make sure I understood how Wikidata functioned. And then I wanted to explore the data itself. So since I had this input sheet from ClinGen database, I took that, and this is a very messy set of figures, but just an exploratory analysis from my understanding of R. And you can look at this yourself if you'd like, where you can see kind of, and then I think this is also a GIF that I put here. So this is looking at the before and after of basically the before and after representing the criteria that I was trying to figure out of how I wanted to filter for what constituted in those 600 entries or so, what constituted as something that could be useful and something that that is not high enough quality or integrity for us to integrate into Wikidata. So then here's a little GIF that I made with R, just something cute, but it's a pretty messy figure, but just to show kind of where there might be biases in the data, either in the gene or disease level. And then also just like my own exploratory analysis. And so I have this process here, and this is my own, notice that this is my own personal GitHub. And then what we put in the scheduled bots is what actually ends up being loaded. So if we go to scheduled bots, and we go to ClinGen, it's just these very clean 2 files, the notebook, the kind of most recent or final notebook, and then the Python script. And whereas here, there's a bunch of different files, and this is kind of my own notebook process and workflow process, and continuously trying to be good about updating those. Okay, so now I've kind of manually tried things out. I've explored the data in the ways I can. So I usually did R. I knew R pretty well at the time, and now I do more Python. So going forward, I would do things in Python and keep everything in Python. And then starting to do tests of the Python script. So if we go to this site, so this is again one of the folders in my own personal repository. So you can see the process I have from September to November. and the different stages. So it's, you can see, this is the very first notebook, and I hadn't really done a lot with Jupyter Notebook until then. And so you can see Andra helped me with a bit of input. I had not even used Wikidata Integrator before, so you can see there's just a lot of things where I'm just kind of copying and pasting and not really knowing what's going on. Here's the QID, here's a property, I think for that association, gene association, and then here's my way to log in. And then you can see how different this is in the November 7th version, where you can go ahead and I encourage people to check out the drafts of this, but you can see now I have these things of checks or, tests or assertions, things that I still need to do. And then the main script, so here's all the process. Here's my kind of test set that I was working with to make sure things work. Here's the way I've been filtering things out. And then I have some functions defined. And here's my main for loop that I then end up making into the bot. And I have some pseudocode here of things I wanted to complete and fill out. And so then I got that to work. And then once that was working, that's when I got to the scheduled bots here. And this is the final script. So the notebook, the Jupyter Notebook is really used as kind of your test or your sandbox for yourself to try things and see that things are working, and then iterate through the entire data set once you've confirmed that things are really looking good for that small set. Because once you write something, right, if you're running a script on 5 things versus 100 things, and that's going to end up going to Wikidata, if you make a mistake that you might not see until later, obviously it's going to take a lot more effort for you to go back and fix all of those if it's more than 5 or 10 versus just trying out a little test or a subset data at first, and then getting that feedback from the community, and then making sure and trying the entire data set. And so here's the entire script here that runs. That's what the queue is in Jenkins that you saw when we were looking at the console output. Okay. And so, yes, and so this is, you can see that I was just doing a subset of 15. I think I started with 5, and then I would do 15, and then I would just move things over. So I would look for odd cases in my data from that first exploratory analysis I did. And then from those odd cases, I would try those out and see, kind of debug and troubleshoot as much as possible for then making a more holistic, sustainable script. And a lot of this came thanks to the feedback I received from Andra, Lynn, et cetera, from the Gene Wiki group. Okay, and then iterations from the feedback. So feedback, I can't emphasize enough how valuable it is, how valuable the community is. So here, Python, you can see in the script itself, Andra helped a lot with comments, Andrew helped a lot, Ginger saying also helped a lot, and I think I have a few other names in the final version. But these are the main checks that I had for myself, and then things to kind of continue. So even when you have a running bot and things are working, there might be still things you want to polish. And so there's always going to be a few small things, but you ultimately just want to have that base functional aspect. Is it doing the things you want? And is it making sure that it's not doing anything wrong? If it's doing something wrong, then that's a problem, right? So really working those kinks out as much as possible on the subset of data, and then trying out the rest. And so here are some of the things that I did, that I validated that these worked in my script. And then automating, so this is just kind of the first section of that script. Again, I already clicked on this from before, but you can see here's my notebook, and then here I converted my notebook over to just a full long Python script, and then I tried that out. I ran that first manually on, thanks, Sago. I ran that first manually on my own computer, and then once we knew that that worked on my own computer, then we switched it over to the protein box spot. But of course, as you get more familiar, then you'll get through the process of how much you want to translate that over. Okay, and then overall advantages. So I think the main advantages here are just the transparency. So using Wikidata, using GitHub, and having this all available on GitHub. And maybe I'll fully admit that myself, had not ever done this before. I felt really nervous about the critiques I was going to get, and just like, oh my gosh, everyone's going to think I'm dumb, I don't know what I'm doing. I mean, people really don't care. Honestly, I feel it's more, you're doing a service that's helping the larger community. And so if that interest is there and you care about certain data sets, I mean, that's really, people are going to appreciate that. And like I said, this group, the Gene Wiki group has been really, really encouraging. And, there hasn't been a time where I felt dumb, even if there's been moments where I've I'm sure I've messed up and did something wrong, that I should have done something else. And I think that process has been really critical to then encouraging and kind of incentivizing for this transparency, because, you know, you want to be able, basically Wikidata, we want to be able to set things up so that, you know, 2 years from now, 5 years from now, 10 years from now, if something comes up, we can more easily see what went wrong and we can address that. Just like with the, you know, my bot running and an issue running now, right, that I had, where's that Jenkins link? The issue of this running now, and, you know, being more transparent with my script, it makes it easier to identify where the issue went wrong. And so being as thorough and transparent with your script and your process as possible is not only going to help you when you're looking back and something does go wrong, but it's also going to help the larger community in the sustainability of the contribution of that bot script, or even your own work in the longer term. So definitely encouraging transparency, even though it can feel very bare, and you know, encouraging people to comment and encouraging to have that process be visible either through GitHub and or Wikidata of how that process looks over time and what the different changes are. And that it's totally okay to expect bumps, that you should expect bumps. You know, when I first did this in November, I thought of 2019, I thought, great, I did it, I'm done. You know, I don't have to look at this again. And you know, that's a nice thought, but also it's kind of a bad thought, because, you know, it's assuming that nothing is going to change, and things are definitely going to change. And so, you know, like I said, you can see here that I did this revision in January, February, because of the change with the, the change with the amount of data and the columns in the input data of ClinGen going from 9 to 10, and then now it's again from 10 to 9. And in this revision, you know, I didn't just change it to be stuff from, okay, now let's just set up the parameters to read in 10 instead of 9. I also made a bunch of additional updates that I was meaning to do in that same revision. And now again, you know, here we are, September 2020, about a year later, and again, it's giving a problem, and that's totally fine. And also following up. So just because something works, it doesn't mean it did it the right way. So every couple of months or so, I check on these output files to make sure that they look good and nothing weird is happening too, because you can have these blue dots, or you can have something run and it could be doing something totally weird and wrong, and you don't want that either. So just kind of a summary of, you know, I'm continuing to learn a lot. I feel like Lynn and Andra are definitely experts here, and I'm sure they would also say that they're continuing to learn a lot through the process. So it's constantly evolving, and to not ever feel, you know, like you're a novice or an amateur, and that's kind of the benefit of having this community mindset. And so one of the questions that came up were the applications of this and the impact of this. So here are 3 main examples related to biomedical data. So here's Wikigenomes, Clambase, and Cytoscape as a Wikidata extension. And then there's also, for people that might do more R things, there's a plugin for the R that I haven't had a chance to explore yet, but I'm definitely curious for anyone that has. So these have been really impactful projects that have come out of people utilizing Wikidata for this purpose of integrating their data into Wikidata and then getting useful things out of that. And then an extension from that is the Sparkle queries, which I think was brought up in both of the breakout rooms, where then you can get to a stage where you have enough of these different types of data relations to different types of data and associations, and you can start asking questions and answering questions that you can't do just in those data sets alone through Sparkle, and kind of connecting lines that otherwise wouldn't even be possible or wouldn't otherwise even exist. So that's kind of the longer term impact. And with this specific example, there's ClinGen, but there's also a ClinVar bot that I'm working on, and then some other ones I'm going to start working on. And so the compilation of those different bots and how they have associations between genes and diseases in different ways, I can then start to build and put together Sparkle queries that are based on scientific questions I'm curious about and want to answer, which I can't do without Wikidata. And I guess with that, I'll wrap up. And are there any questions? Or general comments?
Yeah, thanks, Saba. That's really good. So I should put my... Hold on, so you can see me. Yeah, that's a really nice overview, and maybe makes it slightly less scary to start working on the complexes, although I'm not sure about the time aspect I have available. Does anyone get like substantial funding to do this, or do you kind of do it as part of bigger projects and just squeeze a bit of time out of those projects to add the data to Wikidata and write a bot?
My position, so I'm a postdoc with Andrew's group, and then also partially a lecturer, but my position is fully funded to do this work. So I know that there are, so this is through the NIH, and then there's another grant I'm going to apply for, but it's through the parent grant that Andrew has, and we did a supplemental. So there's already funding available, and then I applied for a supplemental, and we got that too. So I was a little extra padding, and then there's another one. So there's a number of funding opportunities coming through the NIH as this becomes more and more important. And then I would defer also to Lynn and Andra to comment on how they've found good funding for these types of projects.
Yeah, our efforts are, sorry, I didn't turn my camera off. Our efforts are through R01 funding. So between GeneWiki and Wikidata and the disease ontology project. We've had 2 rounds of those. And I think Andrew's in his 3rd round of the same R01. So
Yeah,
there's some funding. There's not tons, but there's definitely some.
And there are also funding rounds that are not project driven, but small time fundings, where that coming from the Wikimedia Fund, from the Wikimedia Foundation itself, or going from Wikidata itself. But there are also out of the life science, outside the life science, I'm aware there is also a lot of projects going on in the GLAM space, which GLAM is an abbreviation for galleries, libraries, archives, and museums. So yes, it's scattered. I'm not aware. So it's scattered, but it's definitely going on. But there is also a lot of ad hoc use of Wikidata. We should check with the folks at Wiki Pathways. They are running, I think they're running BridgeDB, which is their identifier mapping service, which is completely running on volunteers. So it's just an extension to ongoing projects. So it's not being labeled as a Wikidata project, but part of research projects going on. And 2 years ago, we had a collaboration with the Reactome team where we wrote some bots as well, but that was also done outside of grant lines. So yes, it's a bit of both.
Yeah, and I think a lot of the translation work is done by volunteer groups too, groups that are engaged out there.
And yeah, and that's something where Andrew had a talk about, actually, let me see if I can find the talk while I'm talking about this. Andrew had a talk recently for Scripps. I'll find it and put it in the chat when Andra wraps up. But it's focused on crowdsourcing for kind of biocuration, and actually Ginger in our group spearheaded a lot of that. And it utilizes a lot, like people are really interested in these types of projects. And also people right now, it's a good time because, well, I don't know if it's a good time, but a lot of people are wanting to learn skills because they are doing a lot of things from home. And so a lot of people are curious about learning programming or how to do these types of things, familiarize themselves with different types of technology. And so why not give them these opportunities to try and exercise that through these types of projects? I think generally people are pretty curious, and so you always have when you put a call out for a project, and I know there's different criteria depending on where you're located. Like in the U.S., we have IRBs for any time you involve people as part of your projects. But depending on the question, but generally you'll have a lot of people that will sign up when you put a call out for something, and maybe you have 10 to 15% of that original group that's really active, and that's totally fine because that's still much more than if you didn't put that call out at all. And now you have people that are genuinely interested and they're learning something and they're getting something out of it personally, and you're also getting a kind of extra help on projects you're interested in. And then that's like a really good kind of two-way street, as well as with all the stuff going on globally in the world right now, it's nice for people to be connected to scientists in that way too.
Yeah, you were saying, I think Wiki Pathways, Egon said, they just got their first grant in many, many years that will actually fund some of this work, and that was piggybacked on a COVID application of some sort. But they've done a lot of stuff by either voluntary or integrating into other projects.
And we should also acknowledge a bit of an angle we actually didn't touch on, which I should maybe briefly cover. What Wikidata is also doing is that it is bringing linked data into the research infrastructure ecosystem. Because, for example, if you look at PubMed, I know that over a decade, various initiatives have been trying to get PubMed into a linked data platform. And now with projects like WikiCite, you see it moving forward. So Wikidata is actually not solving it, but by inspiration, you see, and I covered that a bit in the breakout, that about 2 years ago, it became a lot easier to deploy your own so-called quote-unquote empty Wikidata by deploying the Wikibase. So it's data, but it's also opening up the ways to use linked data in other platforms. I mean, the example that Saba gave on the ClinGen data, where the columns is a problem, and which might be a bit softened if, for example, ClinGen itself would start using linked data, because then you don't have to rewrite those mappings on a constant battle. So it's from it's a data provider, but also a schema provider and a way of building knowledge graphs.
Great. And I think with that, we are on perfect time for closing remarks, Andra. And I do have the poll ready for whenever you'd like people to do that too.
So give me a second. Okay. So going to the end of the time, it was great to have you all out here, and I hope to see you in the near future in the week. Wrapping up, I think we can, I already had this in my initial slide, the Wikidata is started as an extension to Wikipedia and can be wrapped up as what Wikipedia is to text and articles, Wikidata is to data. It works, it has the same as Saba has nicely showed, the same backend with respect to discussion pages, history pages. You can see you can see the total history, edit history of a page. It is completely free. So the license, the Wikidata is available for under CC0. So it means it can be used in any context, even in commercial use cases. Anybody can contribute, both bots and humans. It covers not only the life science, so also museums, whether every kind of human society can be covered in. It's very it's scopeless. It integrates well with the semantic web, so it's stable. It has long-term support and is not dictated by funding cycles. But it's incomplete. But that's not a problem. It doesn't need to be. We're not trying to create the final solution. But since it integrates well with the semantic web, we can easily leverage links to other linked data sources in the same access and query languages that is already by using federated queries. And I would like to end with this nice XKCD adaptation that says surveys show that we have 16 competing. We should not try to create a 17th, but Wikidata can be the connecting hub or one of the connecting hubs between the 60. I wouldn't say Wikidata should be the only connecting hub. Here I have such a federated query. I won't go into details there, but we want interoperability. I will end up with for future reading we recently did a bioprint preprint that is currently under peer review. This is our latest paper in eLife. This goes into describing schemas in a machine-readable format. Well, and by this, I would like to end this presentation and thank you for your time. Some final notes or questions we can answer. If not, I thank you for your time, and I hope to meet you either in person or on the Wikidata ecosystem in some or other capacity.
And thanks.